This episode is about practical applications of plant tissue culture. Plant tissue culture can be used as a powerful tool for understanding the physiological and biochemical problems of plant growths. At the same time, it can be very successfully commercially exploited. Here is Mr. K.C. Shroff. Like many other countries, many Indian industrialists are also getting interested in using plant tissue culture products, and we would like to have his opinion as to why he has entered into this field. Mr. K.C. Shroff of Excel Industries.
As an industrialist, when I now look at the science and technology of tissue culture, I find it has come to an age, and for producing uniform planting material or high yield, uniform maturity and better quality, there is no other alternative for our country. Whether we look at the fruit trees like papayas, bananas, bear, amlas, pomegranate or cashew nut even, the only answer would be tissue culture. We go to the medicinal plants, Dioscorea, Google or Rhodopia, there too, India can then emerge with a very leading position on this. We will be needing timber material. We are to develop a vast area for forestry of the right type, and this is where whether we talk of teak, whether we talk of the Himalayan areas, the poplar, the deodar, the pine, again it is the same thing. Similarly, when we start looking at the saline areas, the drought-prone areas, it is only through tissue culture that we will be able to develop the improved varieties, the genetic purity for multiplication that will be required, that will be suitable for these areas. There, whether we are looking at agave, whether we are looking at other foders, whether we are looking at some of the salinity tolerant oil seeds, it will all emerge through tissue culture. You go to another way. When you look at the issues of energy, there too you will find that the energy plantations of future have to be so uniform, so consistent, again in difficult area, tissue culture will take up. And naturally you will ask that how is it that Excel Industries, which has been for half a century in chemical industry, how are we thinking of it? But we are not just chemical industries. Excel Industries has preferred to remain a pioneer in the issues of national interest. Our rural development, our job generation, our wealth generation that has to occur, in all those areas, it will be through biotechnology, and in biotechnology, the best first breakthrough will occur through tissue culture. We have to produce things that have to be quality, quantity, and that have also to be at a price that the consumer can afford. Excel is that reputation. Again, when I am looking at this whole issue of job generation, of millions of people, educated, semi-educated, just a simple rural people, the tissue culture technology has this vast potential. Whether it will be the issue of multiplication, propagation, the laboratory setup that will be there, or the final planting, plucking, anything, it will be through this tissue culture technology that this whole vast gamut of fruits, fibers, oils, timber, or anything that you look at, tissue culture has the potential to take up this responsibility, and that is why we as industrialists have started very intensely looking into it.
One of the major advantages of plant tissue culture or biotechnology is that it has brought science to the masses. The commercial exploitation of plant tissue culture at the moment are one, micropropagation, the other is development of pathogen-free plants, third is varietal improvement, and production of secondary metabolites from cultured cells.
Pathogen-free plants can be obtained by using apical meristem as explants. These apical meristem should contain the dome of apical meristem plus a set of maximum first 2 leaf primordia. So, the width or the diameter of the shoot tip should not increase 0.1 millimeter and the length should be less than 0.3 millimeters. These zones are inherently free from virus and pathogens, and therefore tissues cultured from them provide pathogen-free plants.
As I said that one of the major applications of plant tissue culture is to get disease-free, pathogen-free plants, especially those plants which are propagated by vegetative methods like sugarcane, potatoes, etc. They carry viruses from one generation to another, from one plant to the another. And in this respect, the tissue culture has been proved very effective because by culturing the plants from apical meristem explants can give us totally virus or pathogen-free plants. And here is a chart showing you some of the successes that we have achieved.
It lists some food crops from which viruses were eliminated through meristem culture. They are banana, cauliflower, garlic, peas, potatoes, sugarcane, yams, cocoyam, etc.
One of the most commercially viable application of in vitro technology is micropropagation. Micropropagation of plant tissue cultured material started with micropropagation of ornamental plants, and till today the ornamental plants, the flowers, etc. have remained the most widely micropropagated plants. With our increasing awareness for having more forest trees, or suppose if a person suddenly wants to have lots of fruit trees, etc., availability of plants has become a problem. To get more tissue cultured plants is one of the solutions for that. And for example, plants like pomegranate or date palms, where the rate of seed germination, percentage of seed germination is very low, their tissue culture has started playing a major role.
So far, 4 different ways of in vitro technique has been applied on a genetical basis for the improvement of crops. That is, first, mutant selection or cell line selection. Secondly, by getting somaclonal variants or mutants. Third, by haploid cultures. And fourthly, by protoplast fusion. And here is a culture system in which mutagenesis program can be applied. For that, first, explant is prepared from haploid, diploid, triploid or polyploid tissues, and from them callus or cell suspensions are generated, or even protoplasts are isolated, or cells are plated. These tissues are then subjected to mutagenesis either by chemical mutagens or physical mutagens. They are then allowed to go for 3 to 4 division cycles, and a mutational expression then shows in the cultures. These can be morphological or biochemical. So, variant isolation is done either by biochemical analysis or by observing the cultures and then separating them out and regenerating plants from them. Thus, whatever progeny is created, they are taken for genetical analysis.
Let's talk about cell line selection or mutant selection. In vitro cultures, we can get induced mutants or even natural mutants. Culture of apical meristem has been found very desirable because they have a strict control over DNA synthesis sequences.
Some of the mutants developed by tissue culture techniques are high-yielding mutants, herbicide-resistant mutants, salt-tolerant mutants, drought-tolerant mutants, amino acid analog-resistant mutants, etc.
Now, coming to the somaclonal variants. It has been recognized that somaclonal variants can be potential source of genetic variability. We get somaclonal variants in our cultures due to DNA amplification, chromosomal aberrations, or transposable gene material, gene elements from one loci to the other. And because of these, the somaclonal variants can have the traits which can be inherited by their offsprings also.
Here is a list of some species from which somaclonal variants were taken and incorporated into varietal improvement. If we look at explants, we find that most of them, or rather almost all of them, are non-apical meristematic in origin. In non-meristemic cells, due to endomitosis and polyploidy, we get more somaclonal variants than what we get in apical meristem derived cells. There is an increasing gradient of polyploidy level from the apex towards the base.
Plant regeneration from haploid or anther cultures by Shipra Guha and Maheshwari has opened a new possibility of varietal improvement using in vitro techniques. In haploid cultures, polyploidy has also been observed, which was due to endomitosis or nuclear fusion at earlier stages of their growth. In China, haploids have been used in plant breeding programs, and about 2 decades ago they've started working with rice cultures and have ended up into very successful results and have got a very high yielding variety and resistant variety of rice. Protoplast isolation, fusion and plant regeneration from fused protoplasts have also added up and helped us in varietal improvement of the crops. We'll be discussing more about protoplast isolation in our next episode, but here I would like to tell that there are certain traits which are multi-gene in characters. For example, development of drought tolerant or saline tolerant varieties, they are not single gene trait. Multiple genes are involved and therefore gene cloning or gene transfer may not work. In such cases, somatic cell hybridization can be used, and that can be used only by taking up the protoplasts. Moreover, there are many traits which are present in the protoplast of male cells which do not participate in fusion or fertilization. For example, cytoplasmic male sterility. Now, this type of characters can be incorporated into varietal improvement only if we take up protoplast isolation and fusion work. But as I have told you, we will be discussing about this in detail in our next episode. From time immemorial, plant has been a major source of chemicals. Plant tissue culture has been used for production of industrially important chemicals. Usually, we do not culture the whole plantlets, but we culture the cells which can produce and store the important chemicals, or as we call them, secondary metabolites. These secondary metabolites are not the end products. They are often intermediary products of metabolism, but there has been a very good understanding about their production, the media required for their production, and key enzymes involved in their production. Selection of cell lines having higher yields of these chemicals or the required chemicals have been done, and there are some plants which yield comparatively less amount of secondary metabolites if they are grown naturally, but if these cells or if these plant organs are tissue cultured, their yield goes higher.
For example, glutathione and nicotine are produced in more amounts by Nicotiana tabacum cultures, anthraquinone by Morinda citrifolia, rosmarinic acid by Coleus blumei, ajmalcine and serpentine by Catharinthus roseus, and diasgenin by Dioscorea deltoidea cultures.
A new field of application of plant tissue culture has recently emerged, and that is production of artificial seeds. These seeds are actually somatic embryoids which are developed from the callus cells. The pathway involved in their production is as follows.
First, establishment of somatic embryogenesis, then maturation of these somatic embryos, later on they are synchronized and singulated, then bioreactor production of secondary somatic embryoids, they are encapsulated, encapsulation coating is done and they are dried, and then they are ready for the greenhouse and field planting.
One of the modern applications of plant tissue culture is cryopreservation of apical meristem or somatic embryoids. Now the question is, why do we need a cryopreservation of embryoids? Why not of the seeds? That is because majority of the seeds are not viable for a very long period, and if they are stored for a longer period due to presence of inherent pathogens, they start degrading. Then there are chances that these stored seeds may not be true to the type. There may be character segregation. And another very important criteria is that storage of germplasm seeds can be done only of those plants which have seeds. We have got many plants which have stopped producing seeds because of the continuous use of vegetative propagation. So for such germplasm storage, cryopreservation of tissue cultured somatic embryoids or apical meristems have been found very useful. So we have tried to tell you the various applications of plant tissue culture, applications to the understanding of our fundamental knowledge, and applications towards making tissue culture a commercially viable commodity. There will be problems involved during tissue culture. What are these problems and how to face them? We'll be talking about that in our next episode.